So how do we quantify the interactions between these microbes? That comes as the next interesting question. And how do organisms support each other? So in this study, again it was led by Aarti from my lab, and Professor Smita from Biotech, and Professor Lars Blanc from RWTH Aachen by my collaborators on this. And the question we asked was this: what is the interaction between various kinds of microbes? Okay. And we started looking at how microbes enable each other's metabolisms by their presence. So when you have an organism A, it does something. When you have an organism B in isolation, it does something. But when you get these two organisms together, are they able to better support one another? Are they able to grow better? Are they able to produce new metabolites that they couldn't previously produce because of the presence of the other organism? The idea is simple. We have an index called the Metabolic Support Index, which essentially tries to compute what is the fraction of block or stuck reactions in an organism which are relieved by the presence of the other organism. Right. So when A is by itself, there are so many stuck reactions, but when A is in a community with B, there are so many stuck reactions. And this could be lower. I mean, if it's the same, it's there is going to be no support at all, and it's going to be a 0. And if all reactions that are originally stuck remain stuck, there is no benefit, and MSI is going to be 0. And MSI essentially tries to quantify the extent of benefit in terms of what is the new metabolism that is enabled in a given organism. And you can see that this is clearly an asymmetric kind of index, in the sense A might give very little support to B, but it might take a lot of support from B. Right. So B is very helpful to A, but A is not as helpful to B. So these kinds of predictions or estimations can be made using this MSI index. So let us just take a closer stare at the MSI index. You have these two individual metabolic networks, and you can see that because M10 is not present and so on, R6 cannot happen, which means that M9 and M3 cannot be produced, and R4 cannot happen as well. And similarly here, because M11 is not present, there are a bunch of things that cannot happen here as well. And now, so there are two stuck reactions, R4 and R6 in A, and in B you have a lot more stuck reactions. You have R3, R5, R6 and R8 in B that are stuck. Now you build the community metabolic network, and let us say M9 and M10 are exchangeable between these two organisms, so they get connected in this fashion. And now you see that both these reactions are possible because M10 is available, and this also produces M13 and so on, and R7 has become activated because M9 is available from here, right. So what happens is there is no more stuck reactions in A, but 3 stuck reactions remain in B or organism 2. So MSI of A is 1 because all of its reactions have become unstuck, but B is only 0.4 because only 40% of the reactions, or 2 out of 5 reactions have been relieved by the presence of organism 1 in organism 2. So this is a fairly simple index, and if you see, it's all rooted in graph theory, and we start off with this and try to make predictions. And we looked at this for various known cultures and so on, and we also studied a particular culture in our own experiment, trying to understand various exchanges and so on. And we found that in all of these cases, the organism with higher MSI exhibited a higher biomass in co-culture. So the organism that derives more support grows at a faster rate typically, or so we see. And we also extend this to a few applications. I am going to just quickly go through some of these more advanced applications which start with the concept of MSI and MetQuest and try to build more complex models or answer more interesting questions. So the MSI and MetQuest are essentially our building blocks for answering a lot of complex questions. And this work was led by Cheng in Professor Niranjan Nagarajan's group at A-Star Singapore, and Aarti was also the lead in this study from my side. And the question that is being asked here is, well, when you take antibiotics, it really damages the gut microbiome, but how long does this recovery take? And we do know that it varies from individual to individual, but it was found that there is a specific group of organisms that are highly present in certain individuals and they accelerate recovery. So this is the question. So can you identify and understand why these recovery associated bacteria are able to work so well and resuscitate the gut of individuals after antibiotic treatment, right? So what happens? This is a nice cartoon that A-Star folks made to explain this. So you have a antibiotic administration in the gut, which is basically like a forest fire, and following forest fire, the ecosystem slowly recovers to normal health. So how does the ecosystem in the gut recover to normal health post antibiotic treatment? And it turns out that these recovery associated bacteria seem to have a big role to play here. And what we did was, we again constructed a large number of metabolic networks, community metabolic networks for various pairs of these recovery associated bacteria, and computed what are their interactions, and we found that the recovery associated bacteria derived greater metabolic support from each other than from other non-recovery associated bacteria. So these organisms work well as a group. And it is also possible for us to analyze specific metabolic exchanges, and that is something that I will talk about in the next study. And this is a nice picture that tells you what is the network of interactions, and this one interaction was very interesting for us to study because it was one of the stronger interactions, and this is all the top 10% of the interactions are shown here, the interactions that had higher MSIs and so on, and you see that the node size captures the number of incoming edges and things like that. And this particular example of bacteroids Theta iota omicron and Bifidobacterium adolescentis was also studied in an in vivo model, mouse model, where it was found to aid recovery post antibiotic treatment. Basically, there was a higher microbial diversity in the gut following antibiotic treatment whenever the mice were fed with agavage having beta theta omicron and bacterium adolescentis. So this is one approach where metabolic modeling was able to explain some of the observations that we had and also make interesting predictions for what could be interesting combinations of organisms that can help in the recovery of the gut post antibiotic treatment. We will now look at a deeper insight into the metabolic interactions happening in the gut microbiome itself. We have already looked at the gut microbiome in the context of antibiotic resistance, but now can we understand the normal interactions that happen within a healthy gut? What are the key organisms that drive this ecosystem? There are many interesting questions one can again ask. How do microbes interact in the gut, and how are these interactions affected by the diet, essentially the gut environment which is modulated by the diet? Are there different groups of organisms within the gut that interact differently with one another, like we saw for the RABs? The recovery associated bacteria were a tighter cluster, so to speak. And what kind of metabolites do these gut flora exchange, and how do they contribute to microbial growth and survival and community assembly? And we looked at 52 different species here. So last time we did 21, now we've gone as far as 50, and it's possible to go further too because the algorithm is very scalable, and we made again pairwise metabolic networks. It's also possible to make larger community metabolic networks. This is something we've been doing more recently, and we looked at a few different media. There is a DMEM, which is a representative medium for the minimal gut medium, and then we also looked at a high fiber medium, which is a representative of a healthy gut diet and so on, and how the interactions can vary between these different media. And we looked at both metabolic exchanges and computed metabolic support index between organisms in both conditions. And we found that metabolic dependencies vary with organisms. So we looked at how many bacteria receive help in different conditions from different bacteria. So we grouped them by the phylum. So we have Verrucomicrobia, Proteobacteria and so on, and how they help Actinobacteria, Firmicutes and things like that. Right. So you see that there is Fusobacteria derive a lot of benefit from other organisms in DMEM and some others, and some of these interactions do not change across media, and there are many interesting patterns on how organisms across phyla help one another. And if you compare these two, right, you see that the numbers in the top are a little higher, but some just remain, right, and sometimes you also see more interactions in the high fiber medium and so on. And although the organisms are typically more independent in the high fiber medium, because in the high fiber medium there is generally more nutrition available to the organisms, so they are able to grow more independently and more happily in some sense. And still you do find that there are some organisms that are more dependent in the high fiber medium, which is these are all very interesting hypotheses for us to examine a little bit more closely. So how certain interactions emerge in even high fiber conditions and so on, and how other strong interactions persist across from the minimal gut medium to high fiber conditions. And here again, very interestingly, we looked at centralities. So we found that Bifidobacterium bifidum and Ruminococcus torcus had the highest betweenus centrality in this network of interactions. So this is an association network constructed based on dominant interactions, based on which organism receives or gives more help. And we again find that nearly 2/3 of interactions persist from DMEM to HF, the HF being high fiber. Right. So there are lots of persistent interactions which are very strong interactions between microbes that remain no matter what the medium is and so on. But going past all the biology, some of which might be a little over the top for you at the moment, it is still important to understand that just simple graph-theoretic approaches incrementally built to understand and ask very specific questions of these kinds of systems can give us very interesting insights into the ecology of the gut and the interaction between microbes and how it helps their survival. Right. We were also able to look at amino acid synthesis in the gut. So you have a bunch of organisms that benefit from a bunch of organisms that you see at the bottom with a significant increase in amino acid production. Right. And for example, you see that lactobacilli showed enriched amino acid synthesizing capabilities in the presence of other organisms. So look at this, it's able to produce so many more amino acids in the presence of practically every other organism. And then again a very important aspect, which is the presence of short chain fatty acids. These are essentially very important gut phenotypes. So what is the amount of butyrate or propionate that is produced in the gut, and this has impact on how healthy the gut is and so on. And we could identify what are all the organisms that are producing and are exchanging these kinds of metabolites from one to other, and which are some of these secrete more of these SCFAs and so on. And typically many of these organisms exchange butyrate with many gut flora, and some of the other organisms that you see here exchange propionate. And these short chain fatty acids ultimately are a very important way to orchestrate interactions within the gut. And the point again being that just simplify all of this to the bipartite graph and how certain metabolites are exchanged between those two microbes and how they can actually impact gut health, and you can predict many of them using graph-theoretic approaches. There are some limitations to the kind of approaches we use here, and it is important to always be cognizant of that, because as modelers there are many approaches that are available at your disposal, and you may want to use one or the other depending upon the question that you want to answer and the assumptions that are acceptable and so forth. For instance, we only take a static snapshot of interactions that are happening in the gut, but still the graph-based approaches are very useful and can complement constraint-based models, which typically look at steady state or can occasionally look at dynamic interactions and so on. And the other challenge, of course, is that we make a lot of predictions and we can only validate a part of them given the resources, and which means that there are many more observations that remain to be validated, and we are always looking for interesting experiments in literature which can give us some sort of confirmation for the kind of predictions that we have made. And pairwise interactions are helpful, but we can also look at higher order interactions. This is something that is not too difficult to do, but still involves quite a bit of work which is ongoing. And the MetQuest algorithm itself, the predictions are highly contingent on the quality of the input network, and we don't yet weight the reactions or metabolites and the paths. So are two metabolites that are enabled by the presence of another organism equally important? Some of them are not important and so on. So there are these kinds of questions, and it's also difficult to identify very long pathways, but that's not so much of a limitation because they may not be interesting, they may not be thermodynamically feasible, it might be a path never taken and so on. And lastly, we only look at cellular interactions through the lens of metabolism, and although I have convinced you that that's a very important lens as studied by many people, it still leaves out a lot more that's happening in reality. So these are some things that you need to keep at the back of your mind when you start building these kinds of models of these kinds of systems. So to summarize, we talked about MetQuest, which is a tool to identify metabolic exchanges and biosynthetic pathways, and it's quite useful and scalable and can identify metabolic crosstalks that happen in a community. We also looked at this index called Metabolic Support Index or MSI, which is essentially a metric to quantify what support an organism receives from another in a community. This is quite a generic framework to understand microbial interactions. You can identify metabolic exchanges in a community and you can try to get mechanistic insights, right. So this organism tries to help this organism in this condition by producing this metabolite. So these kinds of hypotheses or questions can be set up and answered using this framework. And I hope I have also convinced you that metabolic interactions in the gut can actually drive community structure and health. This is something that is well known, but we can now look at more mechanistic reasons for these kinds of things happening. Okay. And more broadly, our graph models can help answer many kinds of questions. So what are the metabolic capabilities? What are the metabolic capabilities of an individual organism or a community? There are many studies in which we have looked at this, including our most recent work in Microbiome, which talks about the interactions between microbes in the International Space Station, which is a very highly regulated, hermetically sealed environment, but there are still a few microbes that are sourced from various places, including the astronauts themselves who carry a lot of them. And what are the metabolites that are exchanged between these organisms, and how do organisms depend on one another, and are there higher order dependencies in terms of community level interactions and so on. This becomes very interesting because many organisms may not play a big role in individual interactions, but maybe they contribute to the community, and the community itself contributes to survival of an organism and so on. And there could be certain unique contributors. These can be identified by doing higher level analysis, where we can build larger networks and leave out a single organism and study it and so forth. So with that, we come to the end of this interesting example of how graph-theoretic approaches can be highly useful to study microbial communities. Okay. And we will continue with other examples in the next class.